Hi everyone, welcome back, and this time you should have watched the previous video that introduced cell signaling and communication. This introduced several principles that we'll be building on when we specifically talk about two different types of receptors today. In this video, we'll be talking about specifically G protein coupled receptors. So just to review, when does cell communication occur? Anything that you smell, see, touch, or taste, so the five senses, anytime a hormone is released in one part of a body, you'll have to receive that at another location in the body. Immune system cells need to move towards sites of injury, and they need to know where infections are in the body. Even single-celled organisms need to move towards a food source. So in previous chapters, when we talked about the cytoskeleton, we looked at amoeboid movement and how they form pseudopodia. So they have to use some signaling to know when there's a food source outside. So when you're thinking about when cell communication occurs, you can really think about this happening just about anything that goes down, anything that you respond to. So just to think about the different parts of signal transduction, remember that the first event that we will see is going to be the actual reception. So most of the receptors that we'll talk about today are going to be present in the plasma membrane. So they sort of straddle this world where there's extracellular fluid or outside the cell and the cytoplasm inside the cell. The reception is going to happen when a signal molecule, which I'm going to largely refer to as the ligand, binds to the receptor. So most of the time, as mentioned in the previous video, these ligands can't directly get into the cells themselves. So they need this receptor to kind of communicate what's going on outside. So it's going to be this receptor's job to sort of yell into the cell, hey man, here's what's going on out here. So once this ligand binds, pretty much everything that happens after that, up until the larger scale response, is going to be transduction. So some signal transduction pathways can be short, just a few molecules. Some of them can be really complex, and some of them can activate many different types of molecules and proteins along the way. And then the response. So the response we're sort of thinking of as the large scale, sort of what's going to happen. Is the cell going to divide? Is it going to move towards something? What exactly is it going to do? So that's what we think of as the response, and we'll see some examples where you can actually have multiple responses inside the cell so that the cell can have sort of a coordinated action when something needs to occur. So let's get started with a specific type of receptor called G protein coupled receptors. So this may sound like a really confusing name, but actually it's very straightforward. So G protein coupled receptors are simply receptors that associate with something called a G protein, which we'll talk about in the next couple of slides. So G protein coupled receptors have a structure that consists of 7 transmembrane domains. So you can see the way that they sort of weave in and out of the membrane. An external ligand binding site. So this will be sort of the site of reception event. And an internal site for binding to the G proteins that will associate. So you can kind of see those steps, the structure of it, where the ligand will bind, and where the transduction will initially start inside the cell. So there are many types of G protein coupled receptors, and they're involved with a lot of different functions. So this can include things like vision. So actually, rhodopsin, which is very important for vision, is a G protein coupled receptor. Things like taste, smell. Your immune system has a lot of these G protein coupled receptors. The endocrine system. So you can think about even all types of hormones. This is going to be actually the type of receptor that we're going to see for adrenaline or epinephrine. Neurotransmission. So they're involved with that as well. So again, really anything that you can think, probably a G protein coupled receptor, maybe involved somewhere. So as you might imagine, Because there are so many different types of receptors, different functions, there are a lot of different ligands that can activate these receptors. So each receptor will recognize its own specific ligand. So you can imagine now how many different types of ligands and how many different types of receptors there are. So types of receptors that will recognize certain ligands, we'll see that they recognize odors, again, neurotransmitters, hormones, like epinephrine that we talked about in the previous slide, even chromophores or molecules that respond to light. So we've seen the structure of this. This is the receptor itself. The receptor is not a G protein. The receptor is just that, a receptor. So we do need to talk about the G protein. So the G protein, you can see here, here's our receptor. I've kind of faded it out just to keep it from distracting us. The G proteins are going to be the specific part of this transduction pathway where we can start to get extra specific about what happens inside the cell. So just after it's inside the cell. So this will be our first step after the actual receptor is going to be activated. So there's a lot of different G proteins, but there are 4 main types that people like to talk about. Sometimes they have subcategories as well, but they're categorized primarily by what enzyme they're going to interact with after they become activated. So we haven't talked about that yet, but we're going to get to it. So what they do after they're activated, and whether the enzyme that they interact with, they're going to activate it or inactivate it. So that's one of the categories, and sometimes categorized by a certain type of cellular processes that they're actually going to be involved with. So let's take a look at this. We know what G proteins are. These G proteins are going to be composed of 3 subunits. We have alpha subunit, the beta subunit, and the gamma subunit. So for the most part, when these G proteins are not active, they're all going to be associated together. And so because there are 3 of them, and they're all different, alpha, beta, gamma, these G proteins are also going to sometimes be referred to as heterotrimeric G proteins. So different trimer, that's going to be 3 different 3 G proteins. So I'm probably not going to use the term heterotrimeric G proteins too often, just because I like to take shortcuts when I talk. But if you see this, this is an acceptable name as well. Okay, so we have kind of the structure of the G proteins. So we know what they are. These are going to be guanine nucleotide binding proteins. So that's why they're called G proteins. So where have we heard of guanine before? So guanine is actually the same kind of guanine that you can find in your DNA, but this is also GTP. So we've heard of GTP before. Specifically in the citric acid cycle, we've talked kind of in detail about this. So the fact that this is a G protein means specifically that they can bind to GTP. But here's our GTP molecule. Being bound to a GTP molecule, and you can see it specifically on the alpha subunit here, where it will occur most of the time, having a GTP, a triphosphate, bound here is going to be the definition of an active G protein. So it looks active, we have like kind of a starburst. This is going to turn on our G protein. Once this is active, we'll see in the next few slides that we can actually have some of this G protein move away from the receptor and towards whatever enzyme or what have you it's going to activate. But for now, we can see just that it's active. And eventually, when it is through signaling, it will hydrolyze that GTP to GDP, diphosphate, so we'll lose a phosphate there, and this is back to an inactive protein. So that's what we'll be seeing on the next couple of slides. So let's look at this process where we activate the G protein and think about what it's going to do next. Okay, so here's the overview. We're going to study these panels kind of one by one, but what we're going to be seeing here, again, are the events we're going to have an inactive system here. We're going to introduce our ligand. We're going to see how the receptor becomes active, thus activating the G protein, and what the G protein is going to do kind of in response to this. So let's start. We're going to start with just panel 1, which will be our sort of resting state. So in the off state, there are a few things that we can see here. So first of all, the G protein coupled receptor is not bound to a ligand. Okay, so this is empty, so this is off. You can see that the G protein is bound to GDP, so it's not active. And notice it's not associated with this receptor in any way. It's just kind of hanging out nearby, sort of partially in the membrane. Okay, so this guy's not active. So the next thing that we can see is that we're going to activate this. So let's bring a ligand in the next slide. So this is panel 2. Here we have our ligand, and again, this can be a lot of different things. I like to use the example of epinephrine just because we've talked about this in lab before, but there, again, this could be any number of things. Notice now the changes that have happened here. Okay, so you can see immediately we've kicked out that GDP, goodbye, and brought in a fresh new GTP, which means that this is active. So when our ligand is bound, our actual receptor, the G protein coupled receptor, changes such that it attracts our G protein. The G protein, which is now affiliated with the receptor, can exchange its used up GTP for GTP. So now the G protein is active. So we see an active G protein, and I talked a little bit about what would happen next, but let's see it. So here's kind of the shadow of that receptor, so you can see we're moving kind of to the right, away from it. Okay, when a G protein is active, the alpha subunit can actually move away from the beta and gamma subunit. So this is going to be the one that's actually going to kind of handle the work that comes next. So this alpha subunit will travel to whatever enzyme it happens to be interacting with. Again, we haven't talked about its options, but we'll get to that part too. At this point, it's just important to understand how a G protein can interact with its receptor. So let's go ahead and take a look at the next step. And you can see here's the original receptor kind of over here. We're not using it now. Here's beta and gamma kind of doing its own thing. We're not worried about it. The alpha subunit is now going to interact with some type of enzyme. So we can see that it's associating with something. Again, it's still kind of a mystery because we haven't talked about what this could be yet. But now it's this alpha subunit that's active is going to be affecting this enzyme somehow, either activating it or inactivating it, and this can be a couple of different options for what this enzyme is. So we don't really know what the response will be. So let's take a look. After some time, notice what's going on here. After some time, the subunit can hydrolyze the GTP that it had to GDP, which we saw when we introduced these G proteins. So when this happens, this inactivates the G protein, the alpha subunit. So it goes crawling back to the beta and gamma subunits, back to its inactive form. You can see here, notice that the ligand is still bound. So in the case that a ligand is still bound, this G protein can actually slide right back over and become activated again and go back and hang out with that enzyme that it was associated with. If the ligand is not still there, if it's gone, then the G protein will stay in this resting inactive form. So let's explore this in a little bit more animated form. Okay, so here we've got the outside of the cell, the inside of the cell. Here's our G protein coupled receptor. This is going to be our ligand binding domain and associate with our inactive G protein once we're able to bind a ligand. And so here's our mystery enzyme that we still haven't talked about yet. So you can imagine a ligand coming in to bind. The G protein will be attracted to whatever biochemical structural change happens to the inside of this receptor. The G protein affiliates. It's able to exchange its GDP for GTP, so it's now active. And it will head over to this enzyme, where whatever this enzyme is, it will have an effect. Okay, so if we've activated this enzyme, we can see that it is super effective. Okay, so what does that mean, super effective? So to understand what the actual effect is here, we need to learn more about what these enzymes could be. So we've kind of looked at the first half of this, so let's now explore what happens with the enzymes. So G proteins, again, are categorized by what they interact with and how they affect it. So two main types of enzymes that they can interact with are going to be one called adenylyl or adenylate cyclase and phospholipase C. So sometimes these enzymes are activated and sometimes they're inhibited by G proteins, depending on the type of G protein that interacts with them. I'm mostly going to focus on the ones that activate these enzymes, just so that we can get a good idea of how these pathways can work. So let's explore these two enzymes one by one, starting with adenylyl cyclase. So again, here we see in the system, here is our G protein coupled receptor. Here's the actual G protein that's active. And the enzyme that had been a mystery so far is now going to be adenylyl cyclase. Okay. So adenylyl or adenylate cyclase is an enzyme that will take ATP and convert it into a molecule called cyclic AMP. So you'll see this abbreviated as CAMP. So the reaction, not important to focus too much on the structures, but I think it makes for a nice visual. This is ATP, or adenosine triphosphate. So what we're effectively going to do is cyclize where this phosphate is. You can see it's now cyclized this. So we've got one phosphate. We release two. So this is cyclic adenosine monophosphate. So again, you're not going to have to memorize any of these structures or anything, but I think that, it makes it easier to understand when you actually can visualize what we're talking about here. So cyclic AMP is an important second messenger. Second messengers are molecules that will act to propagate the signal within the cell. Largely, we're talking about non-protein, non-enzyme types of molecules here. So this is a small molecule that's not a protein, it's not an enzyme. This is a second messenger. We'll see several of those today. So cyclic AMP is produced. So again, the order of this that happened, the receptor is activated. It in turn activated the G protein, which then activated adenylyl cyclase, which is catalyzing the conversion of ATP to cyclic AMP. Okay, so cyclic AMP, again, this may not have a lot of meaning for you right now, but this is a really important molecule that can do a lot of things within the cell. One of the things that it can do is activate other kinases, particularly protein kinase A, and protein kinase A can then go on to continue propagating this further in the cell. This is a kinase, so it's going to transfer phosphates. So the overall response that we'll be looking at here can be kind of variable depending on what type of cell we're looking at. So let's look at a short video of the G protein coupled receptor an adenylyl cyclase pathway kind of more in depth.
A hormone binds a receptor, activating a G protein. The G protein's alpha subunit binds GTP and dissociates. It then binds adenylate cyclase and activates it. Active adenylate cyclase catalyzes the conversion of ATP to CAMP and pyrophosphate. CAMP activates other molecules, such as protein kinase A, by binding their regulatory subunits.
So we could see in the last video the way that the activation is going to occur. Again, there are a lot of different things that our protein kinase A can do. So the second type of enzyme that we talked about that can associate with our G protein was an enzyme called phospholipase C. So phospholipase C is an enzyme that can cleave specialized phospholipids. So this, you can see phospholipid. It's got some modifications hanging down here in the cytosol. So it's kind of got a regular sort of phospholipid structure, but then we've got some hydrophilic parts in the cytosol. So one of the common substrates that phospholipase C can cleave is going to be something called phosphatidylinositol, or 5-bisphosphate, or more affectionately, PIP2. So you can see it here, okay, PIP2, okay. So this is attached in the membrane. So this is the whole molecule. When phospholipase C gets involved, it cleaves this PIP2. You can see right where it cleaves, right here. It leaves in the membrane a molecule called diacylglycerol. This is an important signaling molecule on its own, but we're going to ignore it for now and focus on the other half of this. So the other half of this is a molecule called inositol trisphosphate, or IP3. So IP3 is also a second messenger. Okay, so you can think about a ligand binding, G protein activating. This G protein activates phospholipase C, which then cleaves IP3 off, and this is going to be hydrophilic, so it can diffuse all throughout the cell. So what are the ramifications of letting IP3 diffuse through the cell? So in the first Amoeba Scissors video that you guys should have watched, they talked a little bit about ion channels, and we've talked about them as well in other contexts in this class up until now. So again, IP3 is another type of second messenger, small molecule, non-protein, non-enzyme. Okay. This IP3 can actually be the ligand for some types of ligand-gated ion channels. So specifically, note where this one is. This one's on the smooth ER where we store calcium. So this is a calcium channel. So when IP3 is released, it can bind this calcium channel and allow calcium to come out with its concentration gradient into the cytosol. And this can have a lot of different responses. If we remember, calcium is involved with things like action potentials. So there are a lot of different things that can occur here when this ligand is bound. So you can kind of think of this whole pathway as the ligand being present, say, hey, you need to let calcium into your cytosol. It just has to go through all of this to get there, and then various cellular responses depending on what type of cell. So we've talked about now two different types of enzymes that the G proteins can interact with and what some of the ramifications of that will be. We've also talked about the general structure and the way that G protein coupled receptors will start to transduce our signal. So just to conclude, you can think about, again, in the cell, there being multiple types of G protein coupled receptors that exist within even human body, and sometimes even multiple G protein coupled receptors on the same cell. So sometimes they have to receive different signals. Each one of these is going to recognize its own specific ligand, and it may interact with a specialized type of G protein. So some of these G proteins may share, but some of them might be more affiliated with certain types than others. These are in turn going to activate enzymes. So again, the two examples we talked about are going to be phospholipase C and adenylyl cyclase. And each one of these enzymes can propagate its own cellular response. So this is pretty straightforward. This is pretty much true. But sometimes we can have some crosstalk between different enzymes, different things. We can have sometimes multiple enzymes or one enzyme even going to contribute to multiple responses. So sometimes this isn't always linear, but we can think about really what we're doing here is we're receiving a signal, making sure that it's specific so that we know what to do, but we need to coordinate multiple things at once. So sometimes the cell has to do multiple things at once. So we can use the system to sort of have crosstalk and create a lot of diversity in what the cell is doing. So maybe you do a combination of response C, D, and E. Maybe sometimes you do B and D. Maybe sometimes you do just A, but you can create a really diverse set of responses based off of variation in ligands, variation in G protein coupled receptors, G proteins, and the enzymes that they will interact with. So in our next video, we'll be talking about a second type of receptor, receptor tyrosine kinases.